Hello everyone, and welcome to today's seminar. Today we will be talking about the Sapphire FL Biomolecular Imager, supporting researchers at every step from in vitro to in vivo. My name is Dr. Jade Fee. I am an application scientist with Azure Biosystems, and today I'll be taking you through many of the ways in which the Sapphire FL supports the proteomics research pipeline. I would like to begin by introducing you to the instrument, the Sapphire FL Biomolecular Imager. This is a tabletop, high-resolution scanning instrument with a variety of features we'll discuss throughout this presentation. This one instrument is capable of supporting countless applications along the proteomics research pipeline, from initial in vitro validation with cell lysates or fixed cells, to ex vivo tissue imaging, to in vivo imaging of living animals. And this ability to cover this extensive variety of applications really comes down to the design of the instrument, which I'd like to tell you a little about now. The first component that makes the Sapphire FL so versatile is its detector technology. All imagers, from the camera on your phone to advanced medical imaging instruments, capture images through a detector, and there are many different types of detectors with different strengths and weaknesses. The detectors in our Azure instruments differ depending on the intended applications of the instrument and include SCMOS, CCD, PMT, and APD detectors. Let's discuss some of the major differences between these common detectors and how those differences influence the resulting images. The Sapphire FL Biomolecular Imager has a competitive advantage in detection among instruments in its class due to two design features. The Sapphire FL is a point scanner, meaning that all the laser power is focused only on one point at a time. This is distinct from the line scanning technology found in some other instruments, which instead distributes the laser power across a line. Additionally, the Sapphire FL exclusively relies on PMT or APD detectors for fluorescent and phosphor imaging, while others use SCMOS detectors. These two features combined give the Sapphire FL exceptionally high sensitivity. Let's look a little more closely at the differences between SCMOS and PMT APD detectors to fully understand how these detectors influence results. As we mentioned earlier, each detector has its own strengths and weaknesses. The biggest benefit of SCMOS technology is speed. SCMOS cameras detect light very quickly. This makes these detectors superior when detecting transient signals like we see from chemiluminescence. When signal strength changes rapidly, short imaging times are essential. However, when imaging more stable signals like we see from fluorescence, SCMOS is not the best choice. SCMOS detectors are great for general detection, but are not optimized for specific fluorescent wavelengths. They yield overall lower sensitivity for fluorescence compared to APDs and PMTs, and they perform line confocal imaging rather than point confocal imaging, which results in higher background. Higher background in turn results in lower signal-to-noise ratios. APD and PMT detectors, on the other hand, are optimized for specific fluorescent wavelengths, resulting in higher sensitivity and making them the best detectors for fluorescence. APDs and PMTs allow point confocal imaging, which gives lower background signal than SCMOS line confocal imaging. We will come back to confocal imaging towards the end of the presentation to show you how this feature applies to real-world data. As with any detector, APDs and PMTs do have their drawbacks, the most significant of which being that they are slower detectors than SCMOS, making them a poor choice for chemi detection. The Sapphire FL uses APDs and PMTs for fluorescent and phosphor detection, while using an SCMOS for chemiluminescent detection. This ensures that each experiment is optimally imaged for the highest sensitivity and the highest signal-to-noise ratio. The other major aspect of the Sapphire FL that enables expansive application flexibility is its modular design. The FL in Sapphire FL stands for flexibility, and this is showcased strongly by its modularity. Unlike other instruments in its class, The Sapphire FL hosts user-exchangeable lasers and emission filters. This means you can swap out individual channels to accommodate the exact wavelengths required by your individual experiment in under 2 minutes. Each Sapphire FL optical module is composed of a laser as the excitation source and an exchangeable emission filter. Users can mix and match lasers and filters to fit the exact specifications they need for their preferred dyes. With support of wavelengths between 375 and 900 nanometers, the Sapphire FL covers the UV, visible, and NIR portions of the spectrum. Our currently offered optical modules are listed here on the right, with custom options also available by request. By covering such a broad spectrum, the Sapphire FL ensures that researchers are free to choose the dyes and chemistries that work best for them without being constrained by their imager. It is this modularity that truly sets the Sapphire FL apart as a unique and powerful tool in the molecular biology lab. Now that we have learned about the technology driving the Sapphire FL, let's take a closer look at how this instrument supports the proteomics research pipeline. As we've mentioned, the FL in Sapphire FL stands for flexibility, and nowhere is that more apparent than in its application flexibility. The technology behind the Sapphire FL allows imaging of gels, blots, plates, slides, tissues, whole organs, living animals, and so much more. In the next section of the presentation, we'll discuss in detail how the Sapphire FL supports this vast array of applications, starting with samples that require membranes and gels. We'll start with what is probably the most classic example of an assay that requires imaging of a membrane, the Western blot. Western blots are assays used to confirm the presence and relative abundance of a specific protein in a complex sample, such as a cell lysate. Proteins from a complex sample are separated by size using gel electrophoresis. The proteins are then transferred from the gel to a membrane, which is then probed with primary antibodies specific to the protein of interest. Secondary antibodies conjugated to fluorophores are then used to amplify the signal. Finally, the blot is visualized using an imager. The fluorophores emit light when excited by a light source of a specific wavelength. It is these excitation and emission wavelengths that must be compatible between the dye used in the experiment and the scanner used to image the blot. The Sapphire FL is excellent at imaging fluorescent Western blots and can capture multiplex images with up to 4 channels. Throughout this presentation, the optical modules used to capture the image are shown in the bottom left corner. This blot was imaged using the 488, 532, 685, and 784 standard optical modules. Multiplexing is an essential benefit of fluorescent Westerns over traditional chemiluminescent Westerns. With 4-channel imaging, you can image up to 3 targets plus a normalization control, all in one blot with no stripping or reprobing required. By contrast, because chemiluminescent Westerns depend on detection of white light rather than light of specific wavelengths, they are incompatible with multiplexing. Taking advantage of the multiplexing capabilities of fluorescent Westerns is important for situations where there is limited sample available and ensures that each blot provides the most data possible. Importantly, fluorescent Westerns are also quantitative when a normalization control is included. Let's talk a little more about quantitative Westerns and the importance of normalization. Traditionally, quantitative Westerns involved normalizing the protein of interest to a housekeeping protein in the same lane, the reasoning being that the expression levels of these housekeeping proteins remain constant regardless of the experimental conditions. However, we now know that some housekeeping proteins may be affected by experimental conditions. If using a housekeeping protein for normalization, it is important to confirm that its level remains constant regardless of the experimental conditions used prior to running your experiment. To avoid this step, an alternative method is to normalize to total protein. Total protein levels have been shown to remain constant, making this a reliable method for quantitation. For total protein normalization, all the protein in the sample is stained and imaged. The proteins of interest are then normalized to this total signal for quantitative analysis. With the Sapphire FL, up to 3 target proteins and total protein can be imaged in the same blot, ensuring the maximum possible data may be collected from each experiment. In this example, increasing amounts of HeLa cell lysate were probed for 3 proteins of interest and stained for total protein for downstream quantification of the targets. For a Western blot to be truly quantitative, 2 features are required. The target proteins must be normalized to an internal control, as we discussed, and the instrument used to image the blot must have a wide enough dynamic range to accommodate the entire sample. Let's explore this a little further. First, let's talk about what we mean by dynamic range. Dynamic range refers to the range over which signal detected by the instrument stays linear. In other words, it is the range between the lower limit of detection and saturation of signal. In this example, we show a dot blot with serially diluted steps ranging from 2048 picograms to 8 picograms, which produces a wide range of signal strength. When imaged under standard conditions, the upper 4 steps saturate, as indicated in red. The reported signal levels off at the maximum detectable signal, and linearity and true representation of intensity is lost. Saturated areas of the blot cannot be included in quantitative analysis, as the signal detected no longer accurately represents the true signal. This example demonstrates the importance of a wide dynamic range capable of capturing both very weak and very strong signals on the same sample. For samples with both strong and weak signals, the Sapphire FL has an extended dynamic range feature, abbreviated EDR. When EDR is enabled, the same blot can now be quantified over the entire range of the sample. The ability to capture signals over a wide dynamic range ensures quantitative results from each experiment and demonstrates the importance of using an instrument that can keep up with your science. Here is an example of the same 3-channel blot imaged both with and without EDR. On the left, the blot was imaged with EDR. No bands are saturated, so each can be quantified and further analyzed. On the right, you'll notice many of the bands are saturated, as indicated by the light pink color in the image. With standard capture settings, researchers are restricted to quantifying and analyzing either the weak bands or the stronger bands, but not both, in the same image. For samples with a wide dynamic range, it is important to capture the entire sample in a quantifiable manner. We discussed the importance of multiplex fluorescent westerns in allowing researchers to capture more data with one blot. One specific application of multiplex fluorescent westerns is the detection of post-translational modifications, including phosphorylation. Phosphorylated and dephosphorylated versions of the same protein are indistinguishable by size on a typical SDS page gel used for western blotting. Because of this, the signals from the different versions of the protein overlap and cannot be discerned by a chemiluminescent western blot. Multiplex fluorescent westerns, however, permit quantitative resolution of overlapping targets and can be used in phosphorylation studies. In this example, HeLa cells were treated with increasing amounts of IFN alpha to measure the production of phosphor STAT3 from STAT3 by western blot. As INF alpha levels increase from 0 to 100 nanograms per milliliter, a corresponding increase in signal from the IR 800 channel indicates increasing levels of phosphorylation. So far, we've discussed applications that result in samples on membranes. These assays typically begin with protein separation by gel electrophoresis. Sometimes, an assay does not require transfer from a gel to a membrane, and imaging the sample while still inside a gel is best. Gels are simpler and easier than full western blots and eliminate the need for secondary antibodies. Fluorescent gel imaging is a common application that can be used to image fluorescent proteins such as fluorescent fusion proteins. Such proteins fluoresce without the help of a fluorophore conjugated antibody and can be imaged in gel without transfer to a membrane and subsequent probing. Normalization to total protein is not lost with this method, as fluorescent total protein stains can be applied to the gel and provide a fluorescent alternative to visual stains. Here we show an example of a fluorescent gel imaged on the sapphire as part of a coagulation study published in Chemical Science. 2D DIGE is a technique used to separate fluorescent proteins by more characteristics than is possible by Western blot. The 2D DIGE assay requires that a sample be subjected to gel electrophoresis in 2 dimensions, first to separate the proteins by pi value, and second to separate them by molecular weight. In this example from pharmaceuticals, fluorescently labeled plasma samples were resolved by 2D DIGE and imaged with the sapphire. Comasi staining also requires gel imaging. Comasi is a traditional visible stain used for total protein visualization and documentation. It is often used by QC labs to validate cell lines. Staining gels with Comasi is a quick and easy process, and the resulting gels are imaged using either white light or the 700 NIR channel, as shown here. In this example, purified BSA was serially diluted from 5 micrograms to 0.6 nanograms, then separated by SDS page and stained with Comasi. The image was captured with the 685 standard optical module on the Sapphire FL. We touched on the modularity and inherent flexibility of the Sapphire FL system earlier in the presentation. I would now like to show one example of how this flexibility applies to real-world research. With the Sapphire FL, any laser can be paired with any emission filter, opening the possibilities in terms of unique dyes and chemistries. Listed here are the pairings of our standard optical modules, which each include 1 laser and 1 emission filter. Importantly, each of these emission filters can be easily switched with another to create a unique module consisting of a user-selected laser and filter combination. Custom lasers and filters are also available. One prime example of a situation that requires non-standard excitation and emission pairings are experiments using dyes with large Stokes shifts like the Megastokes dyes from Diomics. These dyes have been specifically designed to allow a large gap between the excitation and emission wavelengths. Traditional dyes typically have a great deal of overlap between the excitation and emission spectrum, limiting the amount of emission signal that can be captured. By separating the two, more of the emission signal may be captured. By separating the lasers from the emission filters, the Sapphire FL can adjust to optimally capture signal from even the most obscure chemistries. In this example, serially diluted HeLa cell lysate was probed with anti-STAT3 primary antibody and secondary antibodies conjugated to the DY481XL Megastokes dye. The 488 standard optical module contains a 488 nanometer laser and a 513/17 filter. The emission maximum of the Megastokes dye is above 600 nanometers and could not be detected well with a standard configuration. Instead, the 624/40 emission filter was easily paired with the 488 nanometer laser for optimal detection of this specialty dye. While we focused mainly on fluorescent applications so far, chemiluminescence is still a popular method of detection for Western blots and other assays. The Sapphire FL offers a dedicated chemiluminescence module for such applications. Unlike fluorescent westerns, chemiluminescent westerns depend on an emission of visible light from an enzymatic reaction for detection. This chemistry traditionally requires a darkroom and X-ray film, but with the Sapphire FL chemiluminescence module, such assays can be detected on the benchtop. The chemiluminescence module is equipped with an SCMOS detector to provide highly sensitive detection of transient signals. The chemiluminescent module is also capable of white light imaging. So far, we've shown examples of assays performed on membranes and gels that can be imaged using the Sapphire FL. We'll now move away from flat samples and discuss assays that are performed in multi-well plates. But before we can begin discussing multi-well plate assays, we need to address the fact that these samples have depth. When imaging membranes, the focal plane of the instrument aligns with the surface of the glass platen, putting the flat sample perfectly in focus. When we start talking about imaging samples with depth, like multi-well plates, we must consider the focal plane of the instrument. In a 96-well plate, the sample sits in the bottom of each well, which is a few millimeters above the glass surface. To achieve crisp images, the focal plane of the instrument must be adjusted to match the plane of the sample. With the Sapphire FL, the focal plane can easily be adjusted to match your sample height with the click of a button, so you can have confidence that, regardless of the height of your sample, it can be optimally imaged. When imaging complex samples with depth, it is important to optimize the focal plane. Z-stack images can be used to determine the best focal plane for your sample. The individual Z-plane images can then be overlaid in the Sapphire FL capture software to create a composite image, or used to create a GIF to show each of the layers individually in one file. We will return to our discussion about the importance of Z-plane optimization when we examine animal imaging later in the presentation. For now, we simply want to highlight the importance and ease of selecting the right focal plane for your sample type. A classic assay requiring the use of multi-well plates is the in-cell Western, sometimes called a cell-based ELISA. In-cell Westerns are essentially Western blots in a 96-well plate format. They offer a few advantages over traditional Western blots, including their high throughput nature, which allows more samples to be analyzed in much less time. These assays work well for situations where protein size is irrelevant to the experimental question. Another significant advantage of the in-cell Western is the ability to detect proteins in situ. While Western blots require cell lysis, in-cell Westerns simply permeabilize the cell and nuclear membranes to allow access to internal structures by antibodies while keeping the structure of the cell largely intact. Here, HeLa cells were serially diluted into a 96-well plate for an in-cell Western assay. Two target proteins, HNRNPK and GAPDH, were detected simultaneously with Azure Cyto total cell stain as a normalization control. The assay was imaged on the Sapphire FL at 100 micron resolution with the 532, 685, and 784 standard optical modules. The composite image is centered, and the individual channels are shown on the right. Normalization controls like Azure Cyto are required for downstream quantitative analysis. In this example, in-cell Westerns examining ERK expression were imaged in the NIR with the Sapphire FL as part of a study involving mosquitoes published in ImmunoHorizons. When possible, it is best to detect weak or low abundance targets in the NIR to ensure highest sensitivity. Another common cell-based assay is the live-dead cell assay using AOPI staining. This is a multiplex fluorescent assay using two dyes that stain differently depending on cell viability. The assay is commonly used to address dose-response questions. In this example, HeLa cells were treated with acridine orange, which stains all cells, and propydium iodide, which stains dead and dying cells, and imaged on a cell counting slide at 10 micron resolution. The Sapphire FL 532 propydium iodide standard optical module is specifically designed to accommodate the maximum excitation and emission wavelengths of propydium iodide in assays like this. These assays are performed in cell counting slides rather than multi-well plates, but require similar optimization of the focal plane for imaging. Gene transfection is a common biology lab technique performed in multi-well plates and used to introduce foreign genes to cells for a variety of downstream applications. This is a common technique in experiments that require fluorescent or other types of reporters. In this example of a gene transfection assay, YFP was introduced to HeLa cells using 3 different commercially available transfection kits. As you can see from the image, the kits were shown to yield different rates of transfection efficiency. The Sapphire FL's 488 YFP standard optical module is designed to optimally detect YFP fluorescence and was used to detect signal from transfected YFP in this experiment. Let's look at another gene transfection example. In this experiment, a liquid culture of cells expressing RFP was imaged on both the LOGO by Spectral Instruments and the Sapphire FL. The Spectral Instruments LOGO is a large, expensive, specialty imaging machine used for in vivo imaging of model organisms. While this instrument excels at in vivo animal imaging, it is not designed to image samples like we see here. On the other hand, RFP expression was readily detected by the Sapphire FL. While the Sapphire FL and LOGO are very different instruments and do not directly compete, this experiment highlights the importance of using the best tool for the job at hand. Even when highly specialized and expensive equipment is available, it may be more appropriate to use a comparatively inexpensive instrument designed for your application. In this example, determining transfection success was quick and straightforward using the Sapphire FL, while drawing similar experimental clarity from the LOGO proved challenging. UV imaging, using wavelengths below the visible spectrum, is commonly required for cell-based assays because of reliance on DAPI as a total cell stain. DAPI is a common DNA stain that enters both fixed and living cells and is excited by light in the UV spectrum. The spectral profile of DAPI is shown at the top of this slide. Most fluorescent scanners do not extend into the UV spectrum, with their lowest wavelengths reaching somewhere in the upper 400s. If a researcher tries to excite DAPI using, for example, a 488 nanometer laser, no signal will be produced by the dye. Instead, using a laser in the UV spectrum, for example, a 375 nanometer laser, produces strong signal from the dye. The Sapphire FL is the only laser scanner in its class that offers excitation in the UV range, allowing visualization of DAPI and other UV-dependent dyes. To demonstrate the necessity of UV excitation for DAPI visualization, an in-cell western of serially diluted HeLa cells stained with DAPI was imaged on the Sapphire FL using both the 488 and 375 standard optical modules. Because DAPI is excited by UV light, signal is not detectable with a traditional 488 nanometer laser. Imaging with the 375 nanometer laser allows for detection of DAPI stain. We've discussed examples of membrane, gel, and multi-well plate assays that can be performed using the Sapphire FL. Let's now explore the visualization of samples on microscope slides. Let's first address the differences between and benefits of imaging a microscope slide with a laser scanner versus a microscope. Certainly, a laser scanner cannot provide the resolution of a microscope. However, imaging microscope slides exclusively on a microscope comes with some difficulties. The process of imaging on a microscope is time-intensive, as only one slide can be imaged at a time. Perhaps more importantly, only one part of one slide can be imaged at a time. This makes documentation and tracking difficult. Imaging multiple whole slides at once using a laser scanner is beneficial for 2 main reasons. The first being that the entire slide can be captured, so researchers can quickly document the entire sample in one image. The second is that large quantities of slides can be rapidly triaged, allowing researchers to relatively quickly determine which slides to take to the microscope for further analysis. The Sapphire FL offers a slide holder accessory that makes rapid triage and whole slide documentation simple and repeatable. The accessory importantly holds the slides above the glass surface, which eliminates the chance of Newton's rings interference artifacts obscuring the resulting images. A slide holder protocol in the Sapphire FL capture software ensures repeatable scans of up to 15 slides at a time. The resulting whole slide images are useful for both documentation and for determining which slides are worth inspecting under the microscope. This example from the Journal of Pineal Research highlights the advantages of taking full slide images even when microscopy may also be required. The images were useful in confirming that ISL2 is only detected in the pineal gland of their experimental animal subjects. The section scans were also useful in macroscopic comparisons of ISL2 abundancy between experimental cohorts. These whole slide images complement their images from the microscope. While not a microscope itself, the Sapphire FL excels at taking high-resolution images up to 5-micron resolution. In this example, a mouse lung tissue slide was probed for 2 target proteins and imaged on the Sapphire FL at 5-micron resolution, providing a detailed look at the structure of the lung and the distribution of the targets within the tissue section. Tissue sections are not the only types of samples that require microscope slides. Microarray formatted slides present the opportunity to image many sample types at once. This microarray calibration slide hosts serial dilutions of Psi3 and Psi5 dyes. The spots are up to 280 microns in diameter and are imaged here at 5-micron resolution, demonstrating the precision that is attainable with the Sapphire FL. Microarray formatted slides are important tools across biology and are specifically useful in cancer biology. Tissue microarray slides containing spots of multiple tumors and matched normal tissues are available to researchers for downstream testing. Such samples can be useful in protein expression studies seeking to compare normal tissue with multiple cancer samples. The format of these slides allows many samples to be tested simultaneously. In this example, a multiple tumor-matched tumor and normal tissue microarray from BioChain was probed for HER2 and P53. Differences in expression of the classic breast cancer oncogenes are evident across the microarray and demonstrate the usefulness in such high-throughput assays. The slide was imaged on the Sapphire FL in the slide holder accessory at 10-micron resolution. Our focus thus far has been on in vitro and in situ assays. Now, let's discuss how the Sapphire FL supports in vivo research with live animal imaging. The Sapphire FL is designed to allow imaging of living animals under anesthesia. It has 5 built-in anesthesia ports for imaging multiple animals at once and provides 4 centimeters of clearance between the glass imaging surface and the lid, plenty of space for a small rodent. In general, in vivo assays are often necessary in health-related research because they offer information that in vitro or in situ assays simply can't provide. For example, in vivo assays are relevant in cell migration studies, preclinical drug testing, and any study that requires tracking of a subject or treatment over time. While many studies will eventually require dedicated small animal imaging equipment, such equipment is highly expensive and many labs may not have direct access to such equipment at all times. The Sapphire FL allows researchers to rapidly screen their animals before moving to a dedicated animal imager. The ability to quickly gain this preliminary data from a benchtop imager can dramatically improve the experimental workflow for smaller labs. While not a dedicated animal imager, the Sapphire FL can capture publishable images of fluorescent tumors in vivo. In this study, mice were injected with RFP-expressing 4T1 cells and imaged at 100 microns after 11 days to allow the tumor cells time to grow. Mouse fur autofluoresces, which can be seen surrounding the core of the tumor. The fur covering and adjacent to the tumor was shaved and further removed with NIR to decrease background. The image in the center shows the torso of a representative mouse under anesthesia and the RFP-positive tumor in the middle. The tumor itself is highlighted in the zoomed-in image on the right with a half-centimeter bar for scale. As mentioned earlier, in vivo studies are required for studies that measure change over time, as in a tumor progression experiment. The mice in the previously described study were imaged at 3 time points to measure tumor progression. At 4 days post-injection, we see only autofluorescence from the shaved tumor site. After 11 days, the tumor becomes starkly visible, and at 14 days, it is even further defined. The scale bars allow for comparison of tumor size across the time points. Earlier in the presentation, we discussed the importance of optimizing the focal plane when imaging samples with depth, like a living mouse. This is such an important step because of the confocal nature of the Sapphire FL. The pinhole aperture in confocal laser scanning allows the detection of only the light coming from the focal plane, eliminating the out-of-focus light from above and below the plane. This optical sectioning capability enhances the resolution and contrast of the obtained images. This is why, when we take a Z-stack of images, each image gives a different signal. This GIF of 3 RFP positive and 2 negative control mice was generated in the Sapphire FL capture software from 100 micron images taken at 1 mm intervals between 0 and 3 mm above the glass surface. The signal from the RFP positive tumors is clearly visible, where the negative control tumors appear black from lack of signal. 4 of the mice were sacrificed and their tumors were dissected and imaged directly on the glass of the Sapphire FL at 11 days post-injection. Again, the RFP positive tumors show bright signal compared to the negative control tumors. Imaging dissected tumors or other tissues can be an important confirmatory step in otherwise in vivo studies. A GIF of the tumors imaged at varying focal planes again demonstrates how heavily the detected signal depends on the focal plane. The RFP positive tumors show strong signal throughout the depth of the tumors, while the negative control tumors show only slight autofluorescence near the edges. We've shown that the Sapphire FL is an exceptional fluorescence scanner that can be used to support a multitude of assays. Importantly, it is also compatible with phosphor imaging. Over time, radioactive imaging has gradually been replaced by fluorescence as fluorescence offers significantly lower environmental and personal risk. However, radioactivity is still required for many experiments as it provides the absolute highest sensitivity. Autoradiography is traditionally performed with film, but phosphor screens are reusable over many experiments and can significantly reduce the cost associated with such experiments. The Sapphire FL optical modules are each designed for top performance for their intended applications. As we discussed at the beginning of the presentation, APD and PMT detectors are each specialized for detection of specific wavelengths. In the Sapphire FL phosphor standard optical module, a PMT detector is used to ensure the highest possible sensitivity. The Sapphire FL is also one of only 2 biomolecular scanners in its class to offer phosphor imaging. In this example, phosphor imaging with the Sapphire was used in a mobility shift assay. The researchers examined the binding of 2 factors in response to IFN beta, IFN gamma, or a mixture of the two at low doses, and their findings were published in Frontiers in Immunology. In another recent study from 2023, phosphor imaging on the Sapphire allowed researchers to study CMG helicase activity in the absence or presence of polymerase epsilon. The gels were exposed to a phosphor screen, then scanned with phosphor imaging on the Sapphire. The findings were published in Nature Communications. Today, we have explored how the Sapphire FL supports researchers throughout the proteomics research pipeline with the ability to image in vitro samples, in vivo samples, and everything in between. From gels and blots, to plates and slides, to living model organisms, the Sapphire FL truly can image it all. With up to 5 micron resolution and the capability to image up to 4 fluorescent channels, chemiluminescence, and phosphor, the Sapphire FL is the ultimate solution for the molecular biology lab. We hope you found this presentation informative. Thank you for your attention today, and happy imaging!